we're going to go straight into it, and we're going to pick it up from where we left. So if you guys recall, on Monday, we started discussing this paper about a late onset risk factor for Alzheimer called RHBDF2, but then the paper calls it IROM2 throughout the article. And what we had seen so far on Monday was that microglial cells that don't have IROM2, either because they were obtained from IROM2 knockout mice or because they were obtained through CRISPR knocking out of IROM2 in cells in culture, can't do two things. They cannot process ADAM17 into its mature proteolytic or catalytically active form, and they cannot secrete TNF in response to an LPS insult, right? So that's an indication that these microglial cells need IROM2 to be microglia and respond to LPS attack, and also presumably do it through ADAM17. So the question that we left it at is, what else, other than this failure to produce ADAM17, what else is changing in these microglia that don't have IROM2? So what the authors did was take conditioned media from cells with or without IROM2, and they used proteomics to see what peptides or proteins were missing or in higher abundance, I should point out, in the culture media of these cells. And we went through these volcano plots, and we discussed how anything to the right of the zero in the x-axis means things that are accumulating in your experimental sample versus your control. Anything to the left are things that are in detriment, or that are less in your experimental sample than in the control, right? And we homed in on these two things, ADAM17, which was in a way expected since we knew that IROM2 from previous experiments affected the full processing of ADAM17. And the one thing that caught the author's eye is the presence of TREM2 among those things that were inhibited when IROM2 is knocked out. And the actual last figure that we discussed in class on Monday was that these decrease in ADAM17 and in TREM2 levels in these IROM2 knockout cells could reflect the loss of the entire protein, either ADAM17 or TREM2, or a decrease in a specific portion of those proteins. And to get to that question, what the authors did was map the peptides that they obtained through tryptic digestion to do the mass spec, and they mapped them to the corresponding structures of both ADAM17 and TREM2. And by doing so, they found that when it comes to ADAM17, all of the peptides that were floating around in the conditioned media map to the propeptide that is attached or associated with the non-functional, non-active ADAM17. And what that means in turn is that when you take out IROM2 from microglial cells, the peptide that is telling you, hey, there is a lot less ADAM17 here, is really coming from the non-active form. So in other words, maybe I misphrased that the only thing that is floating around in that culture media is, as expected, what is present in the propeptide of ADAM17, because the rest of the protein should be associated to the cell membrane and therefore shouldn't be floating around. So the only thing that they saw floating around when it comes to ADAM17 was the propeptide. And for TREM2, the only thing that they saw floating around was the extracellular domain that is shed off the TREM2 receptor by, among other things, ADAM17. Does that make sense? So the combination of these two I keep going back to this because it's one of the most challenging conceptual aspects of the paper. This idea that you see drops in levels of things or accumulations of things, but these things were floating around the culture media, so it matters what it was that it was floating around, right? And what this mapping of the peptides that were measured indicates is that what they were measuring were the right thing. It was the propeptide when it comes to atom 17, which is the only thing that you expect to see floating around, and the extracellular domain of TREM2, which is the only thing that you would expect to see floating around. Why? Because TREM2 is known to be shed by proteolytic cleavage, so it's not that surprising that some of the extracellular domain might be floating around as soluble TREM2. Okay? Does that bring us back to where we were on Monday? Any questions, anyone? The propeptide is what floats around. And the fact that there was a lot less propeptide floating around is an indication that without IROM2, there's going to be a lot less processing of atom 17. Okay. Right? So atom 17 has a full-length pre-active form that includes the propeptide. And then the presence of IROM2 will allow this pre-active form to be processed. Right. And then there's going to be propeptide floating around because it gets cleaved off. Right? When you don't have IROM2, there's going to be a lot less of that floating around propeptide. Okay. And that's an indication that atom 17 is not being fully processed to its catalytic form. Yeah? And that is what they had seen with the Western blots in some of the previous figures. I don't have them here, but they're in the slide deck from Monday. Cool. So they decided to, looking at these soluble TREM2 fragments and how there's less of them floating around when the cells don't have IROM2, is good. It's an interesting observation. But they decided to look at this through some alternative approaches. And they take two of them. On the one hand, they're going to look at the soluble levels of TREM2 through a technique called ELISA. And what they see is in the cells that are wild types, that are in this case, it was the non-targeted controls, meaning that they use CRISPR, but they didn't really target any gene in these cells. There is some amount of soluble TREM2 produced by those cells, and they detected that by ELISA. Whereas either the atom 17 cells, somewhat as expected, since that's the protease that is thought to be the one that cleaves TREM2, nor the cells that don't have IROM2 and therefore don't produce fully active atom 17, neither of those produces as much of the soluble TREM2. The IROM2 knockout cells still produce some, significantly less, but some, which may be an indication that that there may be some. Well, let me turn around. So why do you think there is still some cleavage of TREM2 in the IROM2 knockout cells when we said that IROM2 was necessary for atom 17 processing? Why would you see still some left?
Could there be something else that also cleaves TREM2, like atom 10?
Atom 10, so something else cleaving. But if it was atom 10, wouldn't you still expect to see some here when you take out 17? But I like the line of reasoning. So if you said something else that cleaves TREM2, if you apply that reasoning to atom 17 itself, then you solved it, right? So this idea that there could be something else we can apply to, we took out IROM2. Well, IROM2 obviously contributes to processing of atom 17, but it may not necessarily be the only thing. So we took out IROM2. There may still be something else processing atom 17, and atom 17, that little amount is enough to produce some soluble TREM2. When you took out atom 17, it's gone. And that's perhaps a strong indication, though some might argue that that's not gone, right? But a lot less. So that's a strong indication that the atom 17 is probably one of the main, if not the main, protease that produces soluble TREM2. Did that make sense? So anyway, as you see here, does anyone know ELISA and how it works?
Is it when an enzyme is bound to an antigen?
Antibody. Josh?
Yeah, no, it's just like analyze like a serum specifically.
That's right. And there are several versions of ELISAs. There is sandwich ELISAs and a whole bunch of them. But essentially, it all starts with an antigen that you want to detect. And generally, what you have is what is called a capturing antibody. So those capturing antibodies are often attached to a solid surface, right? And the way that works is you run your sample of interest, in this case it would be the conditioned media produced by these cells, through those wells that have antibodies that are fixed to the bottom of those wells or those tubes. And everything will float around, and if there is antigen present in that sample, in this case it would be the soluble TREM2, it will be captured by the antibody, since capturing antibody, right? And then you wash anything else that was present in those samples. So essentially, you're left only with the antigen and the antibody. And then to do the actual quantification, what you do is you throw in a second antibody that is, as Mireya was suggesting, attached to some sort of enzyme that will cleave an invisible substrate into a visible one, or a visible product. And that's what the ELISA readers will read, just the accumulation of this visible product. So it normally goes from a colorless to a colored product, colorless substrate to a colored product. That's correct. So we're detecting things that are floating around in liquids using ELISA. The other approach they took was to, instead of saying, okay, I'm going to focus on the soluble TREM2 that may have been shed off the cells, I'm going to focus on the TREM2 that stays on the cell surface. If you guys recall, the shedding of the soluble TREM2 is of the whole of the extracellular portion of the receptor, right? Adam-17 is presumably cutting that extracellular portion of the receptor at the base, and the whole thing gets shed off. So the counterpart of looking at how much soluble TREM2 is floating around in the liquid around these cells is to look at how much TREM2 is left in these cells. And when they did that, they see that controls have some amount of the TREM2 present on the cell membrane on the surface, but then any of these variants of either the Adam-17 or the IROM2 knockouts have a lot more, significantly more. So that's sort of the flip side of the same coin. You see a lot less of the soluble floating around, and conversely, you see a lot more of the receptor still bound to the surface. For this technique, they used, or for this experiment, they used a technique called FACS. Does anyone know what FACS stands for and how it works? I hear flow cytometry.
It's like fluorescently attached. It's like,
And then there was another hand. Cell sorting, that's right. So we put it all together. So the key word that you're missing is activated. That's what the A stands for. So yes, flow cytometry is the short handle for it. So it's fluorescence activated cell sorting. And can anyone describe the technique real quick? Uh-huh. Correct. So we have a cell that expresses some antigen of interest on the surface. We use antibodies that are able to recognize that antibody, that antigen, and then the antibody itself is attached to some sort of fluorescence molecule. And what Jessica was saying is you run it through a laser. Shoots the laser, and if there is fluorescence, what happens if there is fluorescence? Sorts them, right? You made that gesture. What does that mean? What does that gesture mean? Sort.
The exact way the machine does it, but I remember being told that if it outputs a certain signal, that it gets sorted in a certain area.
Into a certain area. Josh?
Because of the specificity of the cell numbers on
If you have an antibody that is able to recognize them. So you can sort based on any number of things, as long as you can see that thing. You can never, you won't be able to see glycoproteins based on fluorescence, because glycoproteins don't fluoresce. But if you have an antibody that recognizes only the glycosylated versions of a protein, then you use the same trick as usual. You use that antibody to recognize the glycosylated protein, and then that antibody is fluorescent, and the laser will be able to see it. Did that make sense? Yeah? The one thing that I want to point out, though, it's in the name, right? Fluorescence activated cell sorting. So we're kind of conditioned to think that there is always that sorting mechanism. That the cell that, quote unquote, tested positive to the laser will be physically sent to a separate container. That is very true, and it's very cool, because that way you can basically purify a population of cells that pass the test. But it's actually not the most common use of the technique. In the most common use of the technique, the machine is simply asking, do you have this protein or not? And it's asking that question of all of the cells that go single file through the laser, right? So you put your sample in the machine will create a single file of cells, and it will make them run in front of the laser, and every cell gets queried, if you will. Do you have fluorescent? Are you fluorescent? Are you fluorescent? Are you fluorescent? And then those that answer, yes, I am fluorescent, look, I can shine, then those get counted separately. So when we say fluorescence activated cell sorting, there isn't always that physical sorting into a separate container. Most of the time it's just counting. And hence, that name that Melita was mentioning before, flow cytometry, because all we're doing is using flow technology to count cells. Does that make sense? So in many papers, you will see these famous plots in which there is some sort of square with quadrants, and then you'll see dots in one quadrant or the other, and they're color-coded. And all of that is just counting. You're just counting the cells of various types. You may, but most typically you're not necessarily sending anywhere special. Yeah? Cell sorters, real cell sorters are and they're way more expensive, we're more sophisticated. There are some flow cytometers that will do this sorting, as in I can count how many are fluorescents versus not, that are bench top and in a couple of $1,000s, and you have, okay? So there's a big gap in the technology there. Cool. Bless you. All right, so let's move on. So now they can add to these things that they're pretty confident about, that the loss of IROM2 will produce this accumulation of TREM2 on the surface of the cells. And the next question is this extra TREM2 working? And by the way, the model explaining what's going on is down here. These figures from the paper itself, and it's basically the idea that when there is IROM2, there's going to be a lot of functional Adam-17, and that Adam-17 will cleave the whole of the extracellular portion of TREM2, whereas if you killed either IROM2 or Adam-17, now that TREM2 is intact, and it should be able to work for longer, since it won't be clipped off, right? So if that is true, how would you check? How can you test that hypothesis? What could you do?
How would you do the Western blotting again? And then check.
So the Western blotting, they did, I didn't show it, but they did it. It's in the paper. And the Western blotting will tell you there is more TREM2 on the surface, but it's not really getting at the question, does it work though?
Checking the phosphorylation of the
SYK, this one, this.
Yeah.
Yes, that's exactly what they did, because that is the first functional step from a functional TREM2 receptor. So when the TREM2 receptor is present and functional, you will expect that there is this interaction with the DAP12 receptor, and that's going to trigger a series of phosphorylations, one of which will be the phosphorylation of this adaptor molecule called SYK. It's actually a kinase, so it's the phosphorylation of one of those early kinases that will initiate a kinase cascade inside of the cell. Yeah, that's exactly what they did. So they took, BMDM stands for bone marrow derived, and I can't remember what the M goes for, it's most likely macrophages, and they did that from either wild-type mice or mice lacking IROM2. The first plot shows us that the IROM2 knockouts are indeed knockouts. They don't have any IROM2 in them. The second plot shows us that the mature form of ADAM17 is gone from those mice, as expected. Calnexin is being used as a loading control here, housekeeping gene. But the real plots here are those two down there. So the first one measures the amounts of phosphorylated SYK, right? And it's barely detectable in wild-type cells when you take out IROM2 and presumably elevate the levels of TREM2 because it's not being shed off the surface of the cells. Now there is also an elevation in the amounts of phosphorylated SYK. And why do they have a blot for SYK, the protein here? Why do you think they do that? If they're seeing an increase in the amount of phosphorylated SYK, why ask a question about SYK, the protein as a whole? Gabby?
To show that the phosphorylated one is the one related to the iron knockout and not just the protein.
The protein itself, yeah. Anytime that you want to make any claims about a change in the state of a protein, for instance, SYK is being phosphorylated more, one of the things that you need to demonstrate is that this increase in phosphorylated SYK is not really due to an increase in baseline levels of SYK. Because if it is, then the whole model that SYK is the same, but now it's being phosphorylated more, falls apart. If the levels of SYK, what we would normally call in jargon total SYK, also go up with a lack of IROM2, then, and we need to be careful here, we need to tread carefully here. It's not like there is not more phosphorylated SYK in the cells. There is. When we compare the phosphorylated SYK to, say, calnexin or actin, there is an increase in phosphorylated SYK. No question about that. But mechanistically, this argument that it is because there's more TREM2, and therefore there is more phosphorylation of SYK, that doesn't hold as much water anymore. It could be that the TREM2 that is on the surface, that accumulates on the surface, is not functional, and the reason the cells accumulate more phosphorylated SYK is just because there's more SYK to phosphorylate. Okay? So anytime that you see, and we've seen it in previous papers, probably we didn't pay as much attention, but anytime that you see blots for phosphorylated or acetylated or methylated or this or that protein, then it's often accompanied by a blot for the total protein, just to make sure that the protein being phosphorylated or ubiquitinated or whatever, the total level of that protein remains the same. Yeah? And to the right, there is simply the quantification of these plots, densitometric analysis showing that there is an increase in the phosphorylated SYK. Sounds good? Yeah? Cool. So let me go back here. So then they ask the following question. So cool, you take IROM2, there is less processing and activation of atom 17. There's a lot more TREM2 on the surface of these cells. We knew from figure 1 in the paper that when you take out IROM2, these cells don't produce TNF. That's something that we know from the beginning. But the question now becomes, okay, so they're not producing TNF, yet they are expressing a lot more TREM2. And TREM2 is one of those receptors on the surface that triggers the inflammatory state of these cells. Right? So it's a little weird that these cells are not producing TNF and yet, or responding to TNF, and yet they have a lot more of the pro-inflammatory receptor in them. So they decide to explore this question a little further, and they do an experiment called a nanostring RNA expression analysis. So you can think of nanostring as a version of RNA sequencing, except that instead of asking what RNAs are present in these samples, it basically works on a specific subset of RNAs that you're interested in. So there is two advantages to this nanostring technology and one disadvantage. The advantages are that you can focus on RNAs that may not be all that abundant and that the typical RNA sequencing may just miss. And secondly, you're not looking at perhaps tens of thousands of transcripts all at once. You can focus on the couple hundred that you're interested in. Right? So those are advantages. The disadvantage is that nanostring technology is very, very unique. So you need to get the reagents from a handful of companies, and it can get a little pricey. But at the end of the day, these people figured that it was better to use nanostring than regular RNA sequencing. So what they did is they used nanostring to analyze these 235 microglia activation genes. Right? They homed in on this very specific set of genes associated with microglia activation. And they saw that some, but not all, of the DAM-associated genes were upregulated by the loss of IROM, and some were not. If you guys don't remember, DAM stands for disease-associated microglia, and it's just a name that we give to these microglia that have been woken up by some sort of injury or amyloid beta plaque or something that tells them you gotta go to work and you have to initiate an inflammatory cascade, right? So what the authors suggest, and I don't have any proof for this, but what they suggest is that the loss of IROM2 seems to promote a pre-DAM state, right? There are some genes that are being upregulated, all of the genes shown here, and some genes, a handful of them that are, let's talk about it in a bit more detail. So they say that there is a pre-DAM state because I want you to notice the graph. So the graph is a little different from the previous volcano plot that we saw. In the previous volcano plot, if you remember, there was that curved dashed line that basically determined what genes passed the FDR, the false discovery rate, and which ones didn't. There is no such FDR curve here. So this plot is a little different. Why do you think that is? Yeah, but this is fold change too. So the x-axis is identical, is the fold change between IROM knockouts and wild type. And the y-axis is identical too, is the negative logarithm 10 of the p-value. So the axes are the same. This one doesn't have the dashed FDR curve, the other one did. And why did it need to be stricter? That's right. That's right. It comes down to the number of data points that you're analyzing that may skew your statistics. So here we're talking 235 genes. The statistics that we're going to use for this are pretty robust on themselves. The statistics with these universe of genes that we're looking at, if you set the p-value threshold at 0.05, as we normally do, there is no need for repeated measures corrections. You're asking this question only 235 times. The other one, we're in the tens, if not hundreds of thousands of comparisons between the knockout and the control. And the more you run a comparison, the more that you might find that are statistically significant just by chance. So there is a mathematical correction for that, and that turned out into that FDR curve that they drew in the previous one. So it's just a matter of how many questions you're asking.
So what if there's you're analyzing thousands of those? Would you still do the same?
So then you would, yeah. If you do RNA sequencing, so the mouse genome may have, like the human genome, approximately somewhere between 30 and 50,000 RNA isoforms, right? And you could detect each of those separately by RNA sequencing. So in those cases, it may not be a bad idea to calculate some sort of FDR. Will you see it always in all papers that people do that? Not necessarily, but these guys did. Yeah. So in these volcano plots, there is two things going on here. On the one hand, there is the dots that are gray versus the dots that are red, right? And the dots that are gray are those that did not pass statistical significance on their own. The p-value is over 0.05. So we're not going to really pay too much attention to them. What the authors did here is they labeled some ones that are known to be associated with the dam state. And they're basically telling you, look, these guys we know are associated with the dam state, and they're not changing significantly. Okay? Then there is all of those that did pass the statistical significance, but there is these two very interesting vertical dashed lines at plus 35% and less, or a minus 35%. Those give us an idea of the magnitude of change. So at those levels, anything to the right of plus 35, it means that there was a change in the amount of that particular RNA. There was more than 35%. Anything to the left of the minus 35, it means that there was a decrease in the amount of that RNA that made it less than 35% of what you find in the control. What the authors are basically inviting us to do is to use a little judgment in saying all of these genes change significantly, and that's what statistics tell us. That's fine. So the change is statistically significant. But we also need to ask ourselves the question, is it biologically relevant? Is it biologically meaningful? Is the magnitude of change meaningful? I can, for instance, measure 10,000 humans here, 10,000 humans here for their height. And then I run the stats and I say, yeah, the first group is about 0.70 centimeters taller than the second group. Right? Okay, that's cool. But is it biologically meaningful? Are you going to bet on any money on individuals who are 0.7 centimeters taller than others that they're going to be different in any way? No. Right? So that's essentially what they're saying here is they're telling us what genes passed statistical significance, what genes didn't, and then they're giving us this guidance as to what the magnitude of change was. So CD52, for instance, changed significantly, and there was an accumulation of more than 35% of that gene. But TSPO was significant, but the magnitude of change may have been the handful of percent of what you see in a control, right? So, yeah, statistically significant, but biologically, I don't know how relevant that is.
How would they check, how would they test to see if it's biology?
You have to actually run the test. That's an excellent question. So it's a judgment call. And it's absolutely arbitrary. These lines, we can move anywhere we want, as long as we change that number at the top. You can say, well, I'm going to run my line all the way to, I want changes that are more than 50%. Or any change, 10% or more is enough. So it's totally arbitrary. Is it biologically significant? So then what would you do? Let's say TSPO. right? I personally claim that a change in 10% is not biologically relevant. Then you come in and say, no, of course it is. How would you test it? The question goes for anybody. So the hypothesis becomes, no, even a small change in TSPO is significant, is biologically relevant. How do you test that?
You like take it out or something?
Exactly. So what you would do is you would take cells, knock out IROM2, somehow prevent this accumulation, no matter how mild, of TSPO. So maybe you can find an inhibitor of TSPO function or do a little RNAi, or maybe knock it out altogether. And now what you want to see is that these cells without that slightly larger amount of TSPO are not functioning the same, right? So yes, it becomes like each of these becomes a hypothesis that needs to be tested. When they say plus 35%, minus 35%, it's just an educated guess that if you see something go up by 5% of its original amount, well, maybe the cell can't even tell that difference. Did that make sense? Cool. So looking at this graph, they say, well, yeah, many of the genes, any names that you see here are genes that are known to be associated with the DAM state. So many of the genes that are associated with the DAM state changed significantly, but some of them may have changed in ways that are biologically irrelevant. And some of the genes that are known to be associated with the DAM state didn't even change. Okay? Hence, that conclusion that the loss of IROM2 may be inducing this pre-DAM state. Okay, and finally, what they do is they measure microglial function in its sort of scavenging function directly. So forget DAM state and the genes associated with it, forget TNF expression. They simply ask the question, are IROM2 knockout cells, microglial cells, better at swallowing amyloid beta plaques? And the answer is yes, right? So they stained plaques with a molecule called thiazine red, and you can barely see it with the lighting and everything, but there was a decrease in the red when the microglia didn't have IROM2 in them, meaning that these microglia were better at chewing it up. The graph shows the opposite, shows the plaque clearance efficiency. So the lower the clearance efficiency is, like in wild types, the worse that those microglia are at chewing up and degrading the amyloid beta plaques. The IROM2 knockout cells were better. Why they flipped this up, I don't know. Editorial decision, I guess. And then they also asked, are these microglia better at engulfing up lipids, right? Because we know that amyloid beta, oligomers or peptides or plaques themselves are not the only thing that microglia and macrophages chew up. Sometimes they will take care of things like cholesterol and these things, right? So they wanted to ask if microglia without IROM2 would be better at engulfing and trapping lipids, and they measure lipid droplets using body P, I think they call it. That's the way that is pronounced, body P, a fluorescent molecule that binds to lipids, and they see more of this fluorescence inside of microglia that don't have IROM2. So in conclusion, these microglia are better at engulfing lipids. So we can come full circle, and even though that these cells were not expressed in TNF at the very beginning of the paper, they're actually better at their job as microglia cells. They may not be full-blown DAM state, and that may explain why they're not producing TNF yet, but they're still functioning better than microglia with IROM2 in them. And why is that? Well, because now we think that IROM2 leads to the shedding of TREM2, and therefore reducing the amount of TREM2 available on the surface of the cells, when you don't have IROM2 and you don't have as much active ADAM17, there is an accumulation of TREM2 on the surface that will make these microglia better at things like clearing amyloid beta plaques or engulfing lipids. Okay? Cool. And when it comes to our growing network, what I did is go ahead and add the IROM2 as an activator of ADAM17. We have them coupled here, 10 and 17. I didn't bother separating them, but IROM2 is activating ADAM17. And if you guys remember, I made some emphasis on Monday that when cells didn't have ADAM17, they didn't have IROM2 either. So that spoke to an activating arrow going back to it as well. And ADAM17 in this, we had seen ADAM17 in the context of the LRP1 receptor before, but now we can add this inhibitory arrow to TREM2, meaning that the more ADAM17 active you have in cells, the less TREM2 you will have. Okay? You guys ready for the second paper? Yeah? All right. The second paper posted in Canvas, title Alzheimer's Disease Risk Gene Bin1. Again, we go back to this idea of we find these genes that are associated with developing Alzheimer's disease, but we don't know how they work. In this paper, they look at Bin1 and they claim that it induces tau-dependent network hyperexcitability. So let's take a look. So in the intro, they tell us that Bin1 was one of the first genes linked to AD in early genome-wide associated studies, GWAS, and remains second only to ApoE in genome-wide significance. So even in those early GWASs conducted between people without Alzheimer's and people with Alzheimer's, Bin1 was one of the first ones linked to AD. Importantly, this association has been replicated in data sets with subjects from diverse genetic backgrounds. You may have heard this before, and if not, you'll hear it now. Many of these GWASs were very, very biased for a while. So if GWASs were conducted, say, 10, 15 years ago, they would mostly include very minor subpopulations of humans, right? So it was mostly European populations, or if they were conducted in the US, it was mostly white males. Since then, there's been like a revision and many studies that go back and include more diverse genetic backgrounds and re-ask the same questions. So many of the genes that were associated with, say, Alzheimer's disease in early GWAS studies fell off when you started including more diverse backgrounds, but Bin1 is replicated regardless of background. Another one that makes Bin1 very interesting is that it also emerged as a top hit in epigenetic analysis. So now we're not looking at the sequence of B1 and all of the other genes between non-Alzheimer's patients and Alzheimer's patients. We're actually looking at epigenetic marks. And in this case, it was DNA methylation. So we're not comparing the sequence of genes, we're comparing the levels of methylation of genes. And Bin1, once again, showed up as the top hit when it comes to DNA methylation. All that means is when you look at the Bin1 gene in people with Alzheimer's, you'll see that it is way more methylated than in people without. Okay? And it was observed in preclinical AD patients. All that means is pre-symptomatic. Right? So these are people who later on developed Alzheimer's, and now you can go back to the data set and ask, was there Bin1 methylated back then or not? And the answer was, more often than not, yes, it was methylated. So it's all preclinical. The association between Bin1 methylation and Alzheimer's disease is independent of the genetic variant. So it's not like a given genetic variant of Bin1 is normally methylated, in which case you're barking twice up the same tree, right? So it's not like these Bin1 variant is associated with Alzheimer's. It happens to be hypermethylated. So when I run the epigenetic study, I see the same group of individuals in that category. It was independent of genetic variant. And the usual, you guys can appreciate the usual music in all these introductions. Yeah, all of this evidence about BIN1 and its association with Alzheimer's is very convincing, but the how BIN1 may be related to AD pathogenesis remains unknown. Okay? So what do we know about BIN1, period? Right? We don't know how it might connect to Alzheimer's, but what do we know about BIN1? It turns out that BIN1, all of their isoforms contain a bar domain that mediates membrane binding and curvature. So BIN1 is one of these proteins that will make membranes curve, plus a C-terminus SH3 domain. And SH3 domains mediate protein-protein interactions. In a nutshell, SH3 domains in one protein love to interact with proline-rich regions in other proteins. So anytime they have an SH3 domain in a protein, it is highly likely that they will partner up and interact with proteins that have proline-rich regions. And then neuron-specific isoforms of BIN1 have this CLAP domain that is known to be involved in endocytosis. We don't know anything about BIN1 in the brain, but we do know about BIN1 in the heart. And in the heart, what BIN1 does is it traffics low L-type voltage-gated calcium channels, we're going to refer to them as LVGCCs from now on, into the T-tubules of myocardiocytes, heart muscle cells, and it will make them better at responding and pumping blood by strengthening calcium signaling. So in the heart, it was shown that if these myocardiocytes don't have BIN1, they will have fewer LVGCCs in their T-tubules, and there is less calcium influx and less contraction. What it might do in neurons remains unclear until today. So they approach the question very simply by first overexpressing BIN1 in neurons. They take hippocampal neurons from rats, put them in cultures. These are primary cultures, like we discussed on Monday, and they infect them with BIN1 attached to a fluorescent epitope, these MK2 that they can detect by microscopy, or they use as a control the fluorescent epitope alone without BIN1, right? And then they go ahead and they detect BIN1, the protein itself. They don't go by the fluorescent epitope, they go by BIN protein itself. And they see that in untransfected cells, there is some BIN1, obviously. That's what you would expect. These cells are wild type, so they're expressing BIN1. The cells transfected with the fluorescent epitope alone have some BIN1 in them. One might say, is that more? But we can argue about that later. When you introduce the BIN1 with the fluorescent epitope, clearly there is BIN1 everywhere, right? And by looking at these cells under the microscope, the authors tell us that the BIN1 overexpression did not alter viability of these cells, so they're not dying on them. The morphology is the same, so they have the same structure for soma and dendrites and axons. And they measure the resting membrane potential, so what the membrane potential for these cells are under resting conditions, and those were not affected either. So putting a lot of BIN1 in these cells doesn't do anything to their resting state. But it causes hyperexcitability. So what they did is they culture these cells in these what are called multiple electrode arrays, right? So they have them in culture in very special containers that have in them these little electrodes that will send pulses of electricity, therefore activating the neurons that are nearby, right? So this thing will send a pulse of electricity, and then all of these cells that are near that electrode will respond. Because that's what neurons do, right? They respond to an electrical signal, and then they send their own signal out. So these MEAs can be used to measure the excitability of groups of neurons. And when they did that, they noticed that the cells transfected with the fluorescent tag alone respond at this rate. So every once in a while they'll spike and they'll respond to that impulse. But if they had bin1 in them, they respond a lot more frequently. And these diagrams now here, each of the spikes in the electropherogram, or whatever that is called, I don't know, the activity graphs are depicted as little lines now here. So either by looking at these guys or looking at these guys, what you get is a sense that these cells overexpressing bin1 are hyper-excitable. They're responding to these signals from the electrons, from the electrodes a lot more. And the graphs here on the right show two plots. One is for action potential frequency, so how often these neurons will fire, but also the burst firing frequency. So anytime that there is a group of action potentials, and there are some criteria for calling this a burst, but anytime that you have a group of these, you call that a burst, and not only are the action potentials themselves more frequent in bin1 overexpressors, the bursts themselves are also more frequent. Yeah? Has any of you taken any sort of neurobiology, neuro something? Yeah. Kind of. Here?
Here, Dr.
Banner. Okay. Because this is about the extent of my neurobiology, so enjoy while it lasts.
Can you clarify what exactly is different between action potential and burst firing?
Yeah, action potential is any of these, one line, right? One cell basically letting out an electric current that will move on to the end of its axon and wake up the next. So any of these lines is an action potential. There is some criteria that I'm not familiar with, but whenever the action potentials happen in groups, now we can call this whole thing a burst, right? So just to give you an idea, this cell here had 5 action potentials and 0 bursts. This cell here had 1, 2, 3, 4, 5, 6, 7 action potentials. I just did a 6, 7. A little dated, but still works. And 1 burst. Yeah? You remember, Gabi, in genetics? I had 2, and I went full blown, and it's recorded. Yeah. So again, I'm not a neurobiologist, but I take it that these bursts mean something. So that's why they measure them. Yeah? Cool. So that's fine, but that's on a neuronal population level. These guys are sending electrical signals into a neuronal culture, and they record these these action potentials and bursts from a population of neurons. So it's fine to say that there is a hyperexcitation of the network, but they wanted to see what bin1 overexpression does to a single neuron. And for that, they use something called whole cell patch clamping. So the way it works, and I stole this diagram from the internet. The way it works is you take a neuron, you approach a very fine micropipette. It looks like those micropipetters that you may have used in labs, in the teaching labs or research labs before, but these are super fine. They're cell scale, right? It's really thin micropipettes. And basically you approach it to the surface of a cell. There's the plasma membrane here, the cytoplasm, the tip of the pipette, and you place it gently against the cell membrane, and then you do a gentle suction until you sort of seal the tip of the pipette. And the pipette has sensors that say, I think you sealed it because there's no more flow of liquid here, right? So once you seal it, then there's a strong suction. It's like, and that breaks the membrane, and it basically connects the liquid that is inside of the pipette with the cytoplasm. So now, if you do recordings of differences in potential, you're actually reading what's going on inside of the cell. And you're reading off of one neuron at a time. It's called whole cell clamping because you're reading off of the whole cell. Yeah? There's a similar technique called patch clamping, you may remember from Dr. Ban's class, where you actually measure from a single patch of the membrane. But here we're measuring off of the whole cell. Okay? So what they did is they did whole cell clamping on their cultures, but they wanted to, when they approach with the pipette a given neuron, they don't know if they're approaching what is called an excitatory neuron or an inhibitory neuron. Right? In neural circuits, there are some neurons that are great at waking up the next one and saying, wake up, wake up. And they send that signal and they wake up the next one. There are inhibitory neurons too that do the opposite. They quiet the circuit down. So inhibitory neurons will send a signal to the next cell that says, shh, don't react. It's all good. Yeah? So in the culture, they see all the neurons, but they don't know which one's which. So when they approach with the pipette, they don't know if they're approaching an excitatory neuron or an inhibitory neuron. And if they measure changes in potential, they don't know what that neuron would do to the circuit. If they would wake up the next one or make it, or inhibit it, right? So they have no way to know, but they use this trick. They bathe the cells in a toxin that blocks, for instance, inhibitory GABA receptors. So those are receptors that inhibitory neurons would have. So in a way, they're pharmacologically blocking all of the inhibitory neurons, meaning that if they touch a cell now and that cell is inhibitory, they'll see no change. But if they see any changes, that cell was excitatory. Did that make sense? So they're approaching this population of cells, they don't know which one is which, but they pharmacologically inhibit all the inhibitory ones, therefore they're going to be looking at the excitatory ones. And when they do that, they do this over several cells. Obviously, that means approaching several of those cells in the culture. What they observe is that the spontaneous excitatory potentials, I forget what the SC stands for, excitatory potentials, are diminished when, the event interval is diminished when the cells, the neurons are overexpressing Bin1. What does that mean? That the interval between spikes has been reduced. That's another way of saying these neurons are firing more frequently. The interval between events went down. Yeah? Now it's a little counterintuitive. Again, I'm not a big fan of neurobiologists. They like to, don't tell them, but come on. Just say that the spikes were more frequent. Why do you tell me the inter-event diminished? It's just backwards. But that's how they like to do it. So the inter-event interval was reduced. There's more events in a given unit of time. The amplitude, meaning how deep these spikes were, here's a beautiful example of what we were discussing before in the context of the other paper. Significant reduction in the amplitude of these spikes. And I see Jacob going like, what? Yeah, right? So this was statistically significant. Are you serious? It went from what? From 30 to 29.5? Come on. Right? But they're showing it. That's good. We can use our own judgment. So, conclusion. When excitatory neurons overexpress Bin1, they will fire spontaneously more frequently than if they're not overexpressing Bin1. And the exact same thing they saw with inhibitory neurons. This time they bathe the cells in a drug that blocks the excitatory receptors. So all of those cells went to sleep. They're not going to record anything from them. But they can take a look at the inhibitory neurons, some inhibitory neurons they have in their culture. It turns out that they measured there were way fewer inhibitory neurons than excitatory ones. That's very normal. And of those inhibitory neurons that they could record from, same thing. The inter-event interval was significantly reduced by overexpression of BIN1, and the amplitude was significantly reduced as well. Here, you may notice the graphs, in a way, flip, because there is a significant reduction in the frequency of these spikes, but some might argue, well, again, it went from an event every 150 milliseconds to an event every 125. Is that really much different? But it turns out that not only are these events more frequent, but there was this drop in amplitude that may mean some more. I don't know. I simply don't know. But they're pointing out to us that in both cases, BIN1 overexpressing neurons, whether those neurons are excitatory or inhibitory, they're firing more frequently. They're firing more. Okay? So now they can relate that to what they had seen at the population level. So the hyperexcitation they saw at the population level is probably explained by what is happening to these neurons on a single neuron level. Everybody's a little more edgy in those cultures. Okay? Cool. Make sure that we make it on time. So, then they look at calcium influx into these cells, because calcium influx is ultimately what leads a neuron to communicate with the next. The release of neurotransmitters from a presynaptic neuron to a postsynaptic neuron is mediated by calcium influx. So yeah, we can measure all these action potentials all day long, all we want, but ultimately what's going to determine if a neuron is communicating with the next one or not in a circuit is whether or not there is calcium influx that may induce the release of those neurotransmitters, right? So they introduce into the neurons these fluorescent molecules that will fluoresce only if there is calcium present in the cytoplasm. So these cells have these fluorescent molecule, but they will emit bursts of light as calcium is coming in. And when calcium gets pumped back out, the fluorescent molecule will go back to not fluorescing again. And what they can do is measure these pulses of light out of the neurons. They do that, and they distinguish between three types of cells: cells that are completely inactive, cells that are active but not much, and then cells that are super active, if you will, right? So the percent of active neurons increases significantly when these neurons overexpress BIN1. And the amount of calcium that is going into the neurons, that's what they mean by area under curve, also is significantly increased with BIN1 overexpression. And the number of calcium influx events, which they call calcium transients, also increases significantly. So there is more neurons uptaking calcium. They uptake a lot more calcium and a lot more often when they overexpress BIN1. So this is an indication that the neurons are really becoming more active. They're intaking calcium. So because they knew that BIN1 interacts with this LVGCC in the heart, they asked, could it be that BIN1 interacts with LVGCC in neurons as well? And to remind you, that mouthful stands for blah, 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 calcium channel. So it kind of makes sense that maybe if BIN1 is increasing calcium influx, it may do so by interacting with a calcium channel. So they used a technique called PLA, really cool technique by which you have one antibody targeting one molecule that you're interested in, another antibody targeting another molecule that you're interested in. In this case, one would target BIN1, the other one would target LVGCC. And each of these antibodies are attached to PCR primers. And then you do a little PCR between the two, only if they're close enough to each other. So if these two things in cells are 40 microns from each other or less, then you can generate a little PCR in between the two, and you can detect that PCR using fluorescent probes. Really cool technique. I haven't ever done it myself. So more fluorescent dots means more interactions between the proteins that you're interested in. So when the cells don't have Bin1 overexpressed, there are some interactions between Bin1 and LVGCC. But if you overexpress Bin1, there's a lot more, and that's what's quantified here. Okay? So Bin1 can interact with LVGCC in neurons as well, just like it does in the heart. And they come up with this hypothesis. They say, well, we know that Bin1 interacts with tau. Here's where our friend tau comes in. Because that was shown in a previous paper, and it makes sense because tau has one of those proline-rich regions. Remember that Bin1, among other things, it had an SH3 domain that is really good at interacting with proline-rich regions, and tau has one of those. So Bin1 has been shown to interact with tau in a separate paper. And in fact, multiple studies have demonstrated that Bin1 interacts directly with tau. And then they noticed that LVGCC also has an SH3 domain in it. So they come up with this hypothesis that maybe tau is bridging the two. That the interaction between Bin1 and LVGCC is indirect, that Bin1 interacts with tau through its SH3 versus proline-rich region interaction, and that tau in turn interacts with LVGCC through its own SH3 to proline-rich region interaction. So they suggest that this interaction is indirect. And to sort of support this idea, they basically do a slightly different technique from the one that I just described called BRET. In BRET, what you do is you, again, use very special antibodies, one against tau, and the other one against either Bin1 or LVGCC. And what these antibodies have, instead of having little PCR primers in them, what they have is a fluorescent molecule that won't only emit fluorescence that you can see with a microscope, but will emit fluorescence that will excite the other antibody. So the way this works is if these two things, either tau and Bin or tau and LVGCC, are close enough to one another, then you excite one of the fluorescent molecules, and that fluorescence will excite the other. And then you read the fluorescence from the second, and you will see fluorescence only if these two things were close to one another. So when you only have tau, you normalize the values and you have a fluorescence of 1. That's baseline. But when you either overexpress Bin1 SH3 or LVGCC SH3, now there is increased fluorescence because now these SH3s are indeed interacting with tau and passing on that fluorescence. Did that make sense? I had a question here, but in the interest of time, we're going to skip it real quick. You can, at home later, go check it out, see if you can think about it. So now, the idea is, if tau is mediating the interaction between Bin1 and LVGCC, if I take out tau, I should be able to see that now the interaction between the two is diminished. If the interaction between the two depended on this intermediary in this middle protein, then if I take out that middle protein, I should see that the Bin1 and LVGCC are interacting less. So they go back to PLA, the cool technique with the little primers and the mini PCR going on. And what they see is that in cells that you don't do really much to, you subject them to a scrambled antisense oligo, so you're subjecting them to an oligo that doesn't really target anything. When you don't have a lot of Bin, there is some interaction between Bin and LVGCC, some fluorescent dots. We saw that in the previous image. When you introduce a lot of Bin, there is a lot more fluorescent dots. We saw that in the previous image. But if you introduce an antisense oligomer against tau, both values are reduced significantly. Both the interaction that happens naturally between tau and LVGCC1 is reduced, that's shown here, and the interaction that happens between LVGCC and the overexpressed bin is also significantly reduced. So what that is telling them is that if you take tau out of the way, now bin1, either at its endogenous levels or the overexpressed bin1, can no longer interact with LVGCC as efficiently. And I stress that because if the interaction had completely disappeared, then these two bars would be flat on the ground. But they're not. And real quick, why do you think they may be not flat on the ground? Are we knocking out tau? No, this is just an oligo, an antisense oligo. So antisense oligos, siRNAs, microRNAs, they always take you so far in inhibiting your target. So the argument could be made that this particular oligo is not that great at inhibiting tau expression. So there was some partial expression, partial reduction in expression, therefore a partial reduction in effect. Yeah? So am I gonna make it? Oh, real quick, so they did the exact same experiments but looking at electrophysiology. So these two graphs show what we have seen before. When you overexpress bin, there is more action potentials and more bursts. But if you take out tau now, both of those effects are wiped out. So not only, if you take out tau, not only are you killing the ability of bin1 to interact with LVGCC, you're also killing the ability of bin1 overexpression to hyperexcite a network of neurons. Okay? So we go back full circle with the physiology. So last, I'm going to take a few seconds for this. In the discussion, it says results up there, it should say discussion. They say increasing evidence supports the idea that changes in neuronal excitability may contribute to AD pathogenesis. We had kind of seen that before at the very beginning of the class when we said that activation of the NMDA receptor caused calcium influx, which could activate these kinases that can phosphorylate tau. And remember, phosphorylated tau is bad. It's the toxic form, right? So we had seen that calcium influx can induce phosphorylation of tau, and that's bad for the neurons. Increasing evidence supports that neuronal excitability can contribute to this, and LVGCCs have been linked, separate from bin1 and everything else, to neurodegeneration. And that is most likely because they're inducing the influx of calcium. So more calcium inside of the neurons, more phosphorylation on tau and toxicity. And what this paper is showing is that bin1 helps with that. The more bin1 there is, the more LVGCC that may be letting calcium in, the more that calcium inside of the neuron might be activating GSK3 beta and CDK5, both of which phosphorylate tau, make it form neurofibrillary tangles, making the neuron really, really sick, not work anymore, Alzheimer's. Okay? Apologize for the rush ending, but yeah, with that, I won't, for those of you who came in a little later, I won't hold office hours through the end of the week because I'm away at a conference, but I'll see you on Monday.